Hello guys, I'm Vinith and welcome to the molecular biology series, chapter number 2. Today I'll be discussing about the biosynthesis of the nucleotides. If we talk about the biosynthetic pathways, there are two major pathways involved in the synthesis. So one is the de novo pathway and one is the salvage pathway. So de novo pathway is the synthesis of the purines or pyrimidines from the scrap materials. Okay, like for the pyrimidines, the scrap material will be the carbon dioxide and the glutamine. So from the glutamine and from the carbon dioxide and the glutamine, the entire pyrimidine ring is synthesized atom by atom. Okay, from different sources, and same goes for the purines also. So the entire purine ring synthesized upon the PRPP atom by atom from different amino acid sources. Okay, and also carbon dioxide sources. So this kind of synthesis of the purines or pyrimidines from by atom from the scrap materials is known as a de novo pathway, while the salvage pathway involves already existing nitrogenous bases or NMPs or IMPs and making them into the AMPs or GMPs or UMPs. This process involves the salvage pathway. Okay, so salvage pathway converts the already present nitrogenous bases or nucleotides into adenosine monophosphates or guanosine monophosphates or any other pyrimidine nucleotides. Okay, so this is the difference between the de novo and the salvage pathways. So first we'll talk about the de novo and then we'll go for the salvage path. For the synthesis of the pyrimidines, there are three major sources: aspartic acid, carbon dioxide, and the glutamine. These aspartic acid and the glutamine are the amino acids, and glutamine majorly involves in the amide donor, okay, nitrogen atom donor, okay, and aspartic acid entirely fits in the structure, okay, and the next major important role is played by the PRPP, that is a 5-phosphoribosal 1-pyrophosphate, which will be acting as a pentose sugar in the formation of the pyrimidines or the purines. And there are various enzymes like CPS2, carbamyl phosphate synthase 2 is involved in the pyrimidine synthesis, and transcarbamylase, dehydrogenase, and transferase and the decarboxylase. And there is one special enzyme in this pathway called transcarbamylase enzyme. So that is a single bifunctional enzyme. Okay, so we'll discuss about that also. The first pathway in the pyrimidine synthesis is linking the carbon dioxide with the amide group of glutamine. So this glutamine amino acid donates the nitrogen atom, and when carbon dioxide reacts with this amide group, form the carbamyl phosphate. This carbamyl phosphate also has a phosphate molecule that will be coming from the ATP molecule. Okay, so this reaction also involves the ATP and a divalent cation, magnesium ion, and conversion of carbon dioxide and the glutamine to carbamyl phosphate. Okay, so this reaction is carried out in the presence of an enzyme called as the CPS2, carbamyl phosphate synthase 2, okay, which combines the nitrogen and the carbon dioxide. And the same type of isoenzyme is found in the urea cycle also, CPS1, okay, so which is majorly found in the mitochondria involved in the urea cycle. The major source for the amide nitrogen for this CPS1 is ammonia in case of mitochondria urea cycle, and the main source for amide nitrogen for this CPS2 is the glutamine in pyrimidine synthesis pathway. Okay, the source of nitrogen is also different, and the reaction, the synthetic pathway is also different, and the localization is also different for CPS1 and CPS2. Okay, so there are also certain activators and certain inhibitors for this initial step. So this initial step is considered as the regulatory step in the synthetic pathway. So when there are increased levels of the UTP present inside the cell, they inhibit this pathway. So that means when there is a increased level of the UTP nucleotides, okay, so there would be no requirement of nucleotides further. So these nucleotides in turn inhibit the initial steps of the biosynthetic pathways. Okay, this kind of product inhibition is known as the negative feedback regulation, and that is done by the nucleotides itself. Okay, and this pathway is activated by the PRPP. Okay, well, in case of prokaryotic cell, this pathway is inhibited by the CTP. Now This carbonyl phosphate now join hands with the aspartic acid and form the carbonyl aspartic acid. Okay, so I'll show you the structure how they are formed. Okay, and the enzyme which is involved in the conversion of carbonyl phosphate to carbonyl aspartic acid is aspartate transcarbamylases, which transfer the aspartic acid group, entire aspartic acid amino acid onto the carbonyl phosphate molecule. Okay, then this carbonyl aspartic acid undergoes dehydrogenation, which involves of removal of water molecule. Okay, and forms the dihydroorotic acid. Okay, and this dihydroorotic acid now again undergoes further reduction and forms the orotic acid. Okay, so dihydroorotate dehydrogenase reduces this molecule dihydroorotic acid and converts this into oxidizes this dihydroorotic acid and converts this into orotic acid. Okay, and then this orotic acid in the presence of PRPP, phosphoripysyl 5-phosphate, converts into orotic acid. So this orotic acid is the major precursor for the formation of the UMP. Okay, so this exactly resembles the structure of the uridine monophosphate structure. Okay, so orotic acid. Okay, so this orotic acid now has a ribose sugar attached to it. Okay, and this reaction involves a special bifunctional enzyme known as the orotate phosphoripyl transferase, which is also a decarboxylase enzyme. Okay, so the same enzyme, single enzyme, which has two different functions. Okay, orotate decarboxylase has the transferase activity and also the decarboxylase has activity. Okay, so this enzyme converts the orotic acid to orotylic acid and then orotylic acid to the UMP, uridylic acid. Okay, so when the decarboxylation happens on orotylic acid, it converts into UMP, uridylic acid monophosphate. Okay, and this single bifunctional enzymes, when there are low activities of this single bifunctional enzyme, so that will result in the orotic aciduria. Okay, what happens is these people who doesn't have this enough, who doesn't have enough amounts of the orotate phosphoripysyl transferase or orotate or this enzyme, single bifunctional enzyme, generally orotic acid will not convert into UMP, rather it will get excreted out in form of urine. Okay, so in the urine this orotic acid gets excreted out and that result in the increased acidic levels of the urine and that causes a lot of pain. Okay, so this condition is known as aciduria, and that this aciduria mainly results from the excess formation of this orotic acid. Okay, so these low levels of the orotate phosphoripysyl transferase and OMP decarboxylase will result in the orotic aciduria, and if you supply uridine to the patients who are exhibiting this condition, okay, so increased levels of the uridine will activate this enzyme and convert the orotic acid to uridylic monophosphate. Okay, you can also overcome this aciduria when you supply the uridine nitrogenous bases, uridine nucleosides to the patient. Okay, so this is the actual biosynthetic pathway for the pyrimidines. Okay, and the structures are also given here. So the major source for the CPS2, the major nitrogen source of the CPS2 is glutamine. And this glutamine donates a amine nitrogen to this bicarbonate ion, and this bicarbonate ion utilizing an ATP molecule gets converted into carbonyl phosphate. Okay, which has a phosphate molecule, an amine group from the glutamine, and we also have the exact carbon dioxide group over here. Okay, onto this carbonyl phosphate, entire aspartate molecule comes and joins with the structure. Okay, so to form the pyrimidine ring. Okay, now this pyrimidine ring undergoes the cyclization in the presence of the dihydroorotase, which removes the water molecule. So this oxygen from here, hydrogen ions from here. Okay, so this removes the water molecule, sorry, this hydroxyl group from here, and this not this ketone group. Okay, so hydroxyl group from here and the hydrogen atom from the amine group. Okay, so gets eliminated as water molecule and you will get the dihydroorotate, which is the cyclized form of the carbonyl aspartate molecule. Okay, now this undergoes the dehydrogenation to get converted into orotate or orotic acid, and this orotic acid, when it gets bonded by a phosphoripysyl pyrophosphate, it converts into orotatin 5 monophosphate. Okay, so extra sugar molecule is bonded to this orotic acid, and this orotatin 5 monophosphate exactly resembles the structure of the uridine monophosphate. So the major difference between these two is, one extra carbon dioxide group. Okay, so if we remove that carbon dioxide group from the structure, that will become the UMP. And this formation of the orotate to uridine monophosphate into uridine monophosphate is done by one enzyme, that is the single bifunctional enzyme, that is orotate phosphorybose transferase or decarboxylase enzyme. Okay, so if there are low activities of this enzyme, the patient will exhibit aciduria, orotate aciduria. Okay, the formed UMP will now be converted into UTP or UTP by a special enzyme called as a NUDIKI. I've already said you in the last class. So this NUDIKI is actually a diphosphokinase enzyme, convert the UMP to UDP and UTP, and the form UTP again gets converted into CTP, cytosine triphosphate, in the presence of an amino acid glutamine, which is a amine nitrogen donor, and the presence of CTP synthetase enzyme. Okay, so this enzyme utilizes the ATP. Okay, all the synthetases utilize the ATP, while all the synthesis doesn't utilize the ATP. This is the major difference between the synthetases and the synthesis, remember. Okay, now formed deoxyuridine monophosphate should have to convert into deoxythymine monophosphate. Okay, for this synthesis deoxythymine monophosphate, we require a special coenzyme known as the tetrahydrofolate, which is involved in the one carbon transfer. Okay, so this tetrahydrofolate coenzyme donates its one carbon atom to DUMP and gets converted into dihydrofolate. Okay, so upon this conversion, it also makes this UMP to DTMP. Okay, so in the presence of thymidylate synthase. Okay, so dihydrofolate, the form of dihydrofolate now is now contains only two hydrogens or two hydro groups in this folate enzyme. Okay, so when it utilizes the NADP molecule for its reduction, it again converts into tetrahydrofolate. Okay, now has the four hydro groups. Okay, and this step is also inhibited by a base analog called as a 5-fluorouracil, which forms an intermediate known as a 5-fluorodeoxyuridine monophosphate. Okay, so this is an intermediate which blocks the conversion of UMP to DTMP, and this is majorly done by the 5-fluorouracil, which is a base analog. Okay, so conversion of UMP to DTMP involves the enzyme thymidylate synthase, which requires a coenzyme known as the methylene tetrahydrofolate, which donates a carbon atom to this UMP and converts the UMP to DTMP. Okay, so this is the simple biosynthetic pathway for the pyrimidines. Okay, and the salvage pathway is not much involved in the pyrimidine synthesis. Okay, so but it plays a major role in the purine synthesis only. Okay, so if we talk about the biosynthesis of the purines, okay, so the same thing what we have seen in the pyrimidines, the amino acids involving in the formation of the entire ring. Okay, the same thing happens in the purines also. So a lot of amino acids will be involved in the formation of the purine ring. Okay, like glycine, aspartic acid, glutamine. Okay, and the carbon dioxide from the bicarbonate ions will be involved in the formation of the purine rings. Okay, and it also requires coenzymes and cofactors like formaldehyde tetrahydrofolate or magnesium ion divalent magnesium cation. Okay, and the starting material for the purine synthesis will be the PRPP, so which acts as the foundation for the purine synthesis. Okay, first the PRPP is formed from the ribose 5-phosphate, and then this PRPP will allow the formation of entire purine ring over itself. Okay, so this is the basic introduction. So first we have to activate the ribose 5-phosphate. So the activated form will be the 5-phosphoribosal 1-pyrophosphate. Now this 5-phosphoribosal 1-pyrophosphate will enter into the purine synthetic pathway. Okay, so in order to activate this ribose 5-phosphate, we require a special enzyme called as synthetases, which again requires the ATP molecule in the presence of magnesium ion, convert the ribose 5-phosphate to phosphoribosal 1-pyrophosphate. Okay, that means adding an extra phosphate molecule to it. Okay, and this step is also inhibited by the purine nucleotides itself. When there are abundant levels of the purine nucleotides inside the cytoplasm or inside the nucleus, so there would be no further requirement. For these nucleotides. So these nucleotides inhibit the initial pathways of this synthesis pathway. Okay, so this kind of inhibition, I've already said you, is known as the negative feedback regulation. Okay, and the activators for this step is inorganic phosphate. Okay, if there are abundant levels of the inorganic phosphate inside the cytoplasm, then the synthesis pathway will gets initiated. Okay, the formed PRPP will now enter into the synthetic pathway and join hands with the glutamine, an amino acid. Okay, so this glutamine donates its uh, amine nitrogen to this PRPP by forming a glycoside bond with the amino group. This PRPP gets converted into 5-phosphoribosyl amine. Okay, so this PRPP, okay, allows a amino group to get bonded on this PRPP and converts into what? 5-phosphoribosyl amine. Okay, so now this amine will allow an entire amino acid, okay, glycine amino acid, to get bonded over it. Okay, so when glycine in the presence of a synthetase, okay, so it reacts with this 5-phosphoribosyl amine, it forms the 5-phosphoribosyl glycine amide. Okay, that means entire glycine amino acid is placed over the nitrogen atom. Okay, that forms the glycine amide, and in the presence of a tetrahydrofolate enzyme, which is a one carbon atom donor, which is also a coenzyme. Okay, so this tetrahydrofolate donates a carbon atom to this phosphoribosyl glycinamide and convert this entire structure into 5-phosphoribosyl N-formyl glycinamide. Okay, that means an aldehyde group, formal group, okay, the simple formaldehyde group is getting added over the nitrogen atom, which of a glycine source. Okay, so this transfer of this glycine aldehyde group is done by the coenzyme tetrahydrofolate. Okay, N-formal tetrahydrofolate donates the formal group to the glycinamide, phosphoribosyl glycinamide, and converts this into 5-phosphoribosyl N-formal glycinamide. Okay, now this formal glycinamide in the presence of glutamine gains one extra nitrogen atom, okay, amine nitrogen atom, and converts into 5-phosphoribosyl N-formal glycinamidine. Okay, so the extra nitrogen atom is placed on this carbon. Okay, you can see this nitrogen atom. Okay, now this entire structure will now undergo the cyclization reaction or cyclization in the presence of a synthetase. By utilizing an ATP molecule, the entire structure now gets cyclized into to form a complete imidazole ring. Okay, so this ring is called as imidazole ring. Okay, and yeah, forgot to tell you about the PB analogs. See, these are the para amino benzoxy analog, benzoxy acid analogs. Okay, so these analogs are these analogs resemble the same structure as this formal transferases, okay, or formal transfer the tetrahydrofolates. Okay, and these analogs are given for a lot of bacterial infections to slow down the purine synthetic pathways in the bacteria. Okay, so this folic acid analogs actually resemble the structure of this dihydrofolate reductase or the formal transferases, and inhibit the conversion of formal tetrahydrofolate to tetrahydrofolate. Okay, so they inhibit the one carbon transfer reaction step. Okay, so by inhibiting that step, the entire purine synthetic pathway will get inhibited in the bacteria, and eventually bacteria will die. Okay, so using methotrexate, which is a dihydrofolate reductase analog. Okay, so this antibacteria compounds will inhibit the purine synthetic pathways. Okay, so these are the PB analogs, which has major role in the pharmaceutical industries to control the bacterial infections. Okay, yeah, so getting into the topic, the ribose 5-phosphate, like 5-phosphoribosyl amine-imidazole, okay, now undergoes the carboxylation to attain the fourth carbon of purine ring. Okay, so this COO minus comes from the bicarbonate ion source. Okay, so that donates the carbon dioxide, and this carboxylase enzyme places this carbon dioxide group onto this carbon. Okay, so that makes up the fourth carbon. Okay, so this compound now called as the 5-phosphoribosyl 5-amino-imidazole 4-carboxylate. Because the fourth carbon is now formed, we call that as a 4-carboxylate. Okay, now in the presence of aspartate amino acid, this aspartate amino acid actually donates a amine group to this ring. Okay, but actually initially this aspartate amino acid combines with the entire structure as it is. Okay, and now the structure is called as the 5-phosphoribosyl 4-succinocarboxamide 5-amino-imidazole. Okay, so this structure now contains entire aspartate amino acid. Okay, but this aspartate amino acid actually donates this amino group only. Okay, not the entire structure will be stayed as it is. Okay, so in the presence of an lyase enzyme which breaks the bonds, okay, adenylosuccinate lyase enzyme, this complete structure will get cleaved off. Okay, so the bond that is present between the carbon and nitrogen will now get broken off, and that will result in the elimination of this fumarate molecule, which is a component in the tricarboxylic acid cycle intermediate. Okay, and that will result in the formation of the 5-phosphoribosyl 4-carboxamide 5-amino-imidazole. Okay, we also have the 5-amino group over here. Okay, and imidazole ring over here. So this compound is called as the 5-amino-imidazole compound. Okay, and in the presence of tetrahydrofolate again, this involves getting one carbon atom over the ring. Okay, so that one carbon atom again is donated by the formal tetrahydrofolate, and now the compound is called as the 5-phosphoribosyl 4-carboxamide 5-formamino-imidazole. Okay, nothing but we only have gained one aldehyde group over here. Okay, so this aldehyde group is now responsible for the dehydrolysis, like elimination of the water molecule and cyclization of the ring. Okay, so in the presence of an enzyme, special enzyme cyclohydrolase, which eliminates a water molecule, this oxygen, this hydrogen, and hydrogen from the amino group will get eliminated as a water molecule, and the bond is formed between the nitrogen and the carbon dioxide. Okay, carbon atom. Okay, and the cyclization of the ring takes place to form the inosine 5 monophosphate. Okay, so this structure is a important precursor for the adenosine monophosphate or the guanosine monophosphate. Okay, so this is the entire pathway. You don't have to remember all the enzymes, but you have to remember the sources of amino acids and sources of the carbon dioxide which are involved in the formation of inosine monophosphate. Okay, and where does the cyclization reaction takes place, and the PB analogs. Okay, and the formed inosine monophosphate now converts into AMP and GMP. This is the major conversion thing in biosynthesis pathway. Okay, so this IMP in the presence of adenylosuccinate synthetase, which utilizes a GTP molecule instead of ATP, and also aspartic amino acid, which donates a amino group to this IMP. Okay, get converted into adenylosuccinate molecule. Okay, so this adenylosuccinate synthetase converts the IMP to adenylosuccinate initially. Okay, by utilizing a GTP and aspartic acid. Okay, so this aspartic acid get placed on this carbon, this carbon. Okay, as it is, and then in the presence of adenylosuccinase, which is a lyase enzyme, removes this amino acid. Okay, breaks the bond between this carbon and this nitrogen. Okay, so that the fumarate will get eliminated, as we have seen in the initial biosynthetic pathway, and the structure formed is known as the AMP. Okay, and this AMP negatively inhibits the initial steps. Okay, for its conversion. Okay, when the enough amounts of the AMPs are formed in the cell, inside the cell, so this increased AMP levels will inhibit the adenylosuccinate synthetase enzyme. Okay, so that there will be no further conversion of the AMP to AMP. Okay, so that will be a wasteful process. Okay, so if we have lot of AMP levels, that will be a wasteful thing inside the cell. So that because of that reason, AMP inhibits the enzyme. Okay, and this IMP can also be converted into GMP in the presence of IMP dehydrogenase and the GMP synthetase. Okay, so in the presence of IMP dehydrogenase, this IMP now converts into xanthosine monophosphate, and xanthosine monophosphate in the presence of amino acid glutamine and glutamate GMP synthetase. This glutamate donates an amide group to this xanthosine monophosphate. Okay, and gets converted into GMP, guanosine monophosphate. Okay, so the same way GMP also negatively inhibits the IMP dehydrogenase enzyme. Okay, so this kind of regulation is also called as a negative feedback regulation. Apart from this negative feedback regulation, there is also certain type of drug called as a mycophenolic drug, which is a reversible inhibitor, which is a competitive inhibitor for the IMP dehydrogenase enzyme. Okay, so this mycophenolic acid actually given for this graft rejection patients, for graft transplantation patients. Okay, so what happens in those patients is, so when there is a graft transplantations, the T cells and B cells starts to segregate rapidly. Okay, so in order to stop the T cells and B cells and to stop the graft rejection process, they give the mycophenolic acid. This mycophenolic goes and binds to the IMP dehydrogenase complex in the rapidly dividing B cells and T cells and inhibit their division. Okay, so if we inhibit the B lymphocytes and the T lymphocytes, there will be no activated immune system inside the body, and immune system now is suppressed, and the graft transplantation will be also successful process. Okay, so this is about the biosynthetic pathway, and next one is the salvage pathways. Salvage pathways, I've said you already, whatever the compounds that are actually present in the cytosol. Okay, so these compounds will make the precursors or the actual nucleotides. Okay, so if you look about this hypoxanthine, which is an inosine monophosphate precursor. Okay, so in the presence of HGPRT enzyme, hypoxanthine guanine phosphorybosal transferase enzyme, hypoxanthine gets converted into IMP. Okay, so it gains a ribose sugar and converts into IMP, and again this IMP again converts into GMP and IMP in the presence of different types of enzymes. Okay, so this HGPRT enzyme plays a major role in conversion of the hypoxanthine to inosine monophosphate, and if there are low levels of this hypoxanthine guanine phosphorybosal transferase in certain individuals, that will lead to the Lesch-Nyhan syndrome. Okay, so this is an X-linked recessive inherited disorder. Okay, which lead to the deficiency of this enzyme, HGPRT enzyme. Okay, and this enzyme low activities, or the deficiency of this enzyme results in the formation of results in the decreased levels of the IMPs or GMPs. Okay, and if there are low levels of the GMPs or IMPs, that will be a toxic condition to the individuals. Okay, so that will result in a lot of neurological disorders and also excessive production of the uric acids. Okay, what happens in this pathway is, if the de novo synthesis is inhibited by this deficiency, the salvage the dietary substances, like dietary substance of these nucleotides, whatever the nucleotides gets absorbed from the dietary substances, their level increases in the body, and they get metabolized to uric acids, and uric acid levels increases in the human bodies. Okay, and this uric acid is toxic. Okay, and it also results in a lot of conditions in the body, like pain and all these things. Okay, accumulation of the uric acid crystals in the knees, or in the synovial fluid. Okay, so this condition will be taking place. Okay, and a lot of neurological disorders also happen occurs in the individuals. Okay, so this is about the Lesch-Nyhan syndrome. And then conversion of the formed ribonucleotides into deoxyribonucleotides. Okay, one major problem for the cell is to convert the ribonucleotides into deoxyribonucleotides. Okay, so to incorporate the nucleotides into the DNA strand, we require deoxyribonucleotides. Okay, ribonucleotide is not involved in the formation of the nucleic acids. Okay, DNA. To convert this, a special type of system is involved, that is known as the thioredoxin system. Okay, so this thioredoxin protein is a dimer. It actually presents the dimer. Okay, so when this thioredoxin is present in the oxidized form, so it exists as TSST. Okay, so this is the oxidized form of this thioredoxin. So when it combines with its coenzyme NADPH, it gets converted into reduced form, that is TSH and TSH. So these are the two monomers of the thioredoxin system. So this sulfhydryl groups donate the hydrogen atoms to this ribonucleotide reductase. Okay, so this enzyme takes the hydrogen atom and utilizing 2 hydrogen atom and 1 oxygen from the ribonucleotide diphosphate, this oxygen, it makes water molecule and eliminates the water molecule as it is, and convert this entire ribonucleoside diphosphate to deoxyribonucleoside diphosphate. Okay, so one water molecule will be eliminated, and if you look at the structure, water molecule structure, so it will be like this. So these two hydrogen ions comes from the thyroidexin system, and one oxygen comes from the ribonucleoside diphosphate. Okay, so this is how you convert the ribonucleoside phosphates to deoxyribonucleoside phosphates. Okay, so this is the entire biosynthesis of these nucleotides, purines and pyrimidines. Okay, in the next class we will discuss about the structure and forms of the DNA and properties of the DNA. Okay.